Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Jamie Hall from Intertek Pharmaceutical Services.
Hi, thank you very much. So in this presentation, I'm going to delve into the wonderful world of flow cytometry and explore how we can harness this powerful technique in the regulatory environment. So first in this presentation, I'm going to give you a brief introduction to Intertek and then go into the workings of the flow cytometer itself before I take a look at the applications and some of the challenges such a versatile system can present. So briefly, I would like to introduce Intertek Pharmaceutical Services. Intertek as a whole company is a FTSE 100 company with a network of pharmaceutical laboratories in the UK, US, Switzerland, India, China, and Australia, so we're pretty global. We provide development support through GLP, GCP, and GMP laboratory services, such as bioanalysis, analytical and formulation development, and release testing across all dosage forms. We have expertise in small and large molecule projects and have a vast range of techniques available to support our clients. I am based at our Centre for Excellence in Biological Characterization in Manchester in the United Kingdom. So this slide is just a quick overview of the flow cytometer to get an idea of what we're working with. The inner workings of a flow cytometer vary considerably between instruments, but in general, they're used to measure the physical and chemical properties of cells or particles. By combining various fluorochrome colors and antibodies, a wide range of parameters can be measured in the same population simultaneously. So although it's not strictly part of a flow cytometer, sample preparation is a crucial step that needs to be considered when performing any flow cytometry work. As with most analytical techniques, the adage of rubbish in, rubbish out applies here. To allow samples to be analyzed, they first have to be homogenized into a single cell suspension, then incubated with markers such as fluorescently labeled antibodies to allow identification of the parameters that we want to evaluate. Once sample preparation has been considered, flow cytometers are broken down into three major components. First, we have the fluidic system. This narrows the cell suspension into a stream of single cells to pass through the flow cell, which uses sheath fluid. Next, we have the optics. This consists of a series of lasers which excite the fluorochromes present on the cells, and then we have the detectors, which normally take the form of photomultiplier tubes. A series of specific mirrors and filters allow a broad spectrum of fluorochromes to be detected. The final part of the system is the data processing. As a lot of data is generated in a flow cytometer, this is a crucial part of any system. The relative complexity means that there are a wide range of different systems available with varied fluidics, different colored lasers, and a wide range of detectors. As time has gone by, flow cytometers have become able to identify an increasing number of colors, which has increased the amount of information which can be obtained from a single experiment. However, much of the technology at the cutting edge is designed with early stage research in mind and as such is not necessarily suited to the regulatory environment. Thanks to its versatility, flow cytometry can be used for a broad range of techniques. Here is just a brief list. The mechanisms of pipeline drugs are complicated and varied, and so having techniques that are capable of verifying their working is essential in the drug development process. So one area we're particularly excited to apply flow to is in our cell and gene therapy area. So we're expanding our offering in this GMP testing area, and the ability to immunophenotype cells which are going to be used to deliver therapies is a really crucial is something that's crucial to be able to do. So flow cytometry is one of the most versatile and valuable tools for multi-parametric single-cell analysis. The technology has been used in research as well as in clinical and industrial environments for more than 30 years. More recently, flow cytometry has become an integral component of the drug discovery and development process. Flow cytometry data is useful to support manufacturing as well as safety and efficacy assessments during clinical evaluation. These are critical components of biological license applications submitted to the regulatory authorities. Assays to evaluate functionality of biopharmaceuticals provide confirmatory evidence that they have the appropriate physicochemical properties and give structural confirmation. Validation of these methods to assess critical quality attributes of biopharmaceuticals is a key requirement for manufacturing under GMP environments. The drug development stage will determine if any regulatory standard is required. The different stages also have different requirements in terms of what applications you might want to consider using the flow cytometry for. So, what makes flow distinct from other analysis techniques? Well, the ability to analyze single cells or particles gives you an incredible amount of detail from your assay. For example, if you were to be working with a cell-based assay with a fluorescent endpoint and we're measuring cell fluorescence as an endpoint, if you were to just use a plate reader, it would be unclear if the results that you're observing were due to changes in the entire cell population or just a subsection of the population. With flow cytometry, you can determine if the nature of the response is, for example, just one cell emitting more fluorescence or the entire population shifting, and this gives you much finer detail. Combining this with hierarchical analysis, a specific cell type can even be targeted. You can also measure multiple parameters on individual cells at the same time. For some assays, the noise from dead cells or artifacts could lead to a misinterpretation of the results. But with flow cytometry, these influences can be excluded or even measured if one desires. In general, the life cycle of a regulatory flow cytometry assay begins with a method transfer from another site. In some cases, a technical transfer from another GMP facility may be possible. However, this is often not the case. During this stage, you should assess the differences between the two sites and determine if it is feasible to follow the same approach. In many cases, the differences in instrumentation will mean that it will be better to develop the method again from scratch. The project then proceeds to the development phase, where the method design is investigated and improved upon. The method can be further improved in the optimization stage before proceeding to any validation. It is important that the validation parameters assessed are tailored towards the phase that the project is in. The ICH guidelines should be considered when designing any experiments. So, before going into the hands-on work, I first want to take a step back. Before embarking on any flow cytometry work, it's essential to understand the science behind your assay. One consideration is selecting the appropriate antibody clone to use if you're using antibodies. You need to make sure that the antibody that you're using will bind to the target you're interested in. If you're investigating a drug that binds to receptors you're looking at, it's important to know if they will interfere with the antibody binding. Next, you must consider what fluorochromes to use. A good tip is to use the brightest fluorochromes for your rarest markers. There are a wide variety available, but you must be careful to ensure that you can distinguish between your markers using the system you have available. Using fluorescent spectrum viewers can really help this process. Compensation can overcome some fluorescence overlap, but it doesn't make up for poor panel design. If a sample processing method uses harsh chemicals, you must consider avoiding tandem dyes, as these can degrade, and this will have a huge impact on your results. It's also worth considering appropriate controls. Use of fluorescence -1 controls can reduce the amount of subjective interpretation, and this will really improve validations if the scope for subjective interpretation is reduced. It might also be worth having non-simulated controls to help with your data interpretation. So, during method development, it's important to determine the mechanism that we will be using for cell preparation. For cell lines growing in a liquid culture, this should be relatively straightforward, but adherent cells present more of a challenge. Complex mixtures of cells, such as PBMCs, also present unique challenges. And we should always bear in mind that some cell preparation methods can substantially impact the results. If the drug that's being studied requires cell stimulation in order to function, this should be developed during the development phase. For example, getting the right conditions and the right incubation times is really critical. The order of the steps in the method can also have a substantial impact on results. For example, if you were to be working with adherent cells, you should determine if it is best to stain the cells that touch the surface or to dissociate them prior to analysis. Selecting the appropriate reagents is important, and not just the antibodies. If working with whole blood, the selection of lysis buffer can substantially affect your results. Another important step is antibody titration. When you are developing a method, you should titrate each antibody individually to calculate the stain index for the different dilutions. This allows us to optimize the signal-to-noise ratio and generate much more reliable and consistent results from our assay. It's important not to let your cells die as well. Getting the right balance between obtaining full staining and preventing cell death is crucial to reproducible results. If you're expecting sample acquisition to take a long time, then it might be worth fixing your cells before analysis. So we move into the validation. So typically, during the process of analytical method validation, a variety of assay performance characteristics may be evaluated. These include accuracy, specificity, precision, sensitivity, stability, reference ranges, linearity, and interference. But the intended use of the data and what phase the study is at will dictate which validation parameters need to be evaluated. If, during the lifetime of the assay, the intended use of the data changes, additional validation parameters might have to be addressed. Due to the complexity of flow cytometry and the lack of qualified reference materials, not every validation parameter can be assessed with flow cytometry methods. The format of the assay will determine what parameters you can assess. Assay specificity, sensitivity, precision, stability, and reference ranges can normally be validated for flow cytometry methods. Linearity, standard calibration, and interference can sometimes be validated, but accuracy, selectivity, range of quantification, incurred sample reanalysis, and normal signal distribution can often not be evaluated for flow. When performing a validation, it's crucial to consider the purpose of the assay, as this will determine what parameters you need to assess. I can't emphasize enough how important the method development phase is. If the method isn't right, then you can have real issues in the validation. Before performing the validation, you should know what the endpoint you are looking at is. Establishing acceptance criteria that are appropriate for your assay is also important for a successful validation. These criteria should be tight enough that they meet the purposes of the assay, but not unrealistic based on the data from the development. Again, developing a robust method will help achieve the validated method you need. Another thing that must be considered when performing regulatory analysis is the compliance status of the instrument for electronic records. This is an area that regulators are increasingly looking at in their audits. At Intertek, we use a fax-lyric system which has audit trail capabilities and is fully CFR 21 Part 11 compliant. So one of the greatest strengths of flow cytometry is its flexibility. But working in a regulatory environment, this also presents its challenges. When we consider the flow cytometry itself, all the parameters listed here on the left can be altered, and this must be accounted for in the method. For example, we have the laser power, the flow rate, and also the number of cells that are going to be collected in the assay. Each of the photomultiplier tubes, in our case of which there are 10, can all have their gain adjusted, and this substantially affects the results. When working with a validated method, it's essential that any variable parameters that have an impact on the results are either fixed or subjected to audit trails. The performance of a flow cytometer can vary from day to day. For this reason, it's important to be able to track the performance accurately. Becton Dickinson, who make our flow cytometer, the fax-lyric, provide cytometer setup and tracking beads, often called CsTV beads. These allow the performance of the cytometer to be tracked automatically. We'll come to these beads on future slides. So another challenge is the wide range of instruments available. There are a dozen or so manufacturers making a wide range of models. When you're transferring a method from another site, it's really important to consider this. You must take into consideration the laser and filter setups, and if the fluorochromes will be appropriate for your machine. In some cases, it might be better to start from scratch rather than making compromises, trying to crowbar a method in to fit another instrument. Another area that presents a challenge is the data analysis. So different interpretations of flow cytometry plots by different individuals, or even the same individual on a different day, can drastically alter results. For this reason, it's crucial to have a defined gating strategy in your method that is as objective as possible. To ensure optimum consistent performance of our instrument, the fax-lyric, we use BD's CsTV beads. These beads are fluorospheres with a verified size and fluorescent intensity. When these are run on the instrument, the system automatically adjusts detector voltages based on the expected values. It also allows instrument performance to be monitored over time. If there are any anomalous results, the system automatically alerts the user. The use of these beads ensures the instrument is performing reliably every day. This can dramatically reduce precision issues with the methods. It also means that fluorescence compensation only has to be performed once every 60 days. This saves a huge amount of time and reagents. One other thing to consider with your instrument is having thorough and regular cleaning protocols in place. It may seem trivial, but as a system that uses fluidics, flow cytometers can be prone to blockages if they're not maintained correctly. If these occur during sample analysis, this can have a really, really negative impact on your results. So I've touched briefly on the many ways that flow cytometry data can be interpreted. This leads to issues when working in a regulated environment. Inclusion of gating controls, such as fluorescence -1 controls to set gates, increases the consistency. Again, this is why we must include the gating strategy in the method. One of the ways to reduce the subjectivity is to export the median fluorescence data into another validated software, such as Softmax Pro. This way, the data is interpreted in a much more conventional way with less human subjectivity. This is particularly appropriate if we're looking at cell potency assays. And then if you're using gates which can be adjusted, it's important to have an audit trail. Again, the FACSLyric system provides this. These log when a user makes a change and also require approval before the data can be reported. Whatever the gating strategy or data processing methodology, it's essential that it's part of any validation. If different analysts will be interpreting the data, it might be worth including inter-analyst data processing as part of the validation parameters. This is particularly the case if data processing involves subjective assessments. Wherever possible, the scope for subjective assessments should be reduced, and this goes right back to the importance of the development stage and including appropriate controls in our assays. So, to wrap up, I hope I've demonstrated that flow cytometry is a highly flexible and powerful tool for analysis. But of course, working in a regulatory environment, this can present its challenges. To ensure methods are fit for purpose, their application must be considered, and they must be developed to meet this criteria. Method validation should be thorough and take into account the strengths and limitations of the technique. To ensure that data generated will stand up to regulatory scrutiny, with such a wide range of parameters that can be modified, it is crucial that the system being used has audit trails and is CFR 21 Part 11 compliant. At Intertek, we achieve this with our FACSLyric instrument. It may seem trivial, but it's extremely important that the system maintenance procedures are in place to keep what is quite a complicated system functioning correctly. Again, we use the BD CsTV Beads to make sure our instrument is performing consistently. And in case I haven't emphasized this point enough throughout my presentation, spending on time on method development is crucial for any validated flow cytometry method. Establishing and locking down parameters can really reduce headaches during the method validation stage. So, thank you for listening today. Our contact details are on the screen now. I'll be delighted to hear from you if you have any future questions, but for now, I will hand you back to our moderator for the Q&A session.
Thank you, Jamie. So our first question is, what would be the typical turnaround time for flow cytometry validation?
So if we're working with a fully developed method, a validation would typically take 4 to 6 weeks, and this includes the preparation of validation plan, the analysis, and the report. At Intertek, we can provide an expedited service. This would typically take less than 3 weeks, but it's important to remember that this is assuming we have a fully developed method in place, again going back to the importance of the method development. This method development phase can take a lot longer, depending on how complex the method is, so starting the development early is really important.
Okay. And what flow cytometry assays are you currently running at Intertek?
So all the work we do here at Intertek is performed on behalf of clients, so as you can appreciate, there's quite a few confidentiality agreements in place, so that prevents me going into too much detail. But in the lab at the minute, we have an assay measuring the expression of an internal marker in response to the client's drug. We use medium fluorescence intensity, MFI, as a readout for this, and we export the data to Softmax Pro, and we use this to determine the potency. So this is an assay we'd previously tried to develop using RT-PCR, but found that flow cytometry was a much better endpoint. It's far more reliable. We're also developing an assay for a drug that promotes phagocytosis, and we look at the internalization of a fluorescently labeled protein. So we've got quite a wide range of studies on the go at the minute.
Okay. And what data processing software can we use for regulated data analysis?
So we currently use the Fact Suite software that is included with the Fact Lyric. That's the one provided by BD. We fully validated the system and found that it's perfect for what we use it for, particularly the review and approve functions, which help control the analysis process. There is other data processing software. A lot of people will be familiar with Flojo, and also there's SCS Express. But if you are processing the data for regulatory studies, it's important to ensure that the systems are fully compliant with the regulations. So that goes back to the 21 CFR Part 11 compliance issues again. So it's really important when you're looking at your software to make sure that it's compliant.
Okay. And it looks like we have just time for one more question. So would a regulator expect potency data from flow cytometry and also another orthogonal technique?
So that's a really good question. There are, of course, other means of measuring potency, and the method that you choose can depend on a lot of factors. So whether to include data from one or more approach would depend on the biologics molecule in question, its mode of action, and also the phase of the development. There may be instances where a flow cytometry-based potency assay alone would be enough, but all these factors I've just mentioned really need to be considered. So it's one that definitely we would have to discuss if we were going to look at any methods.
Okay. Well, thank you, Jamie.
Okay, thank you very much.
Thanks to our audience for joining us. The recorded version of this webcast is available for on-demand viewing on our website. As a registered attendee, you'll receive a follow-up email providing us a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you. Please stand by.